Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell. I'm going to be your host here tonight at the Real Science Exchange. Tonight, we have truly a fascinating topic to dive into, the role of mammary gland in governing calcium metabolism. Joining us is a very special guest, Dr. Laura Hernandez, a renowned researcher and professor from the University of Wisconsin. Dr. Hernandez recently delivered an insightful presentation for the Real Science Lecture Series on October 2nd. And you can find that presentation on our website at balchem.com/realscience. And we're thrilled to have Dr. Hernandez with us here today to expand on that discussion. Now, Dr. Hernandez, welcome to the Real Science Exchange. I guess this is your first time to the pub, so welcome.
Thank you.
Any special drinks that you have tonight? I know maybe not so, but if you were at a pub tonight, you got bubbler. If you were in a pub tonight, what would you be drinking?
Probably wine. Probably wine.
Good. Red or white? Do you have a preference?
Definitely red. Definitely red.
Good stuff. I love a good red wine. And I see you brought a guest with you here tonight. Would you mind introducing your guest, and how did you decide to bring him along with you?
Absolutely. Well, I was very honored that Dr. Tom Overton agreed to join us. Tom, I've known since I think I was in grad school, probably read all the papers and have gotten to know him more since I was a professor. And I think I mentioned offline, I am not a nutritionist. I'm definitely a physiologist. So I always like to have a good nutritionist available to chat with. And I think, he's one of the best and definitely the most practical and works in the space I do. And so I think he was the right person to choose to talk about this. So.
Yeah, absolutely. I'll second that. Tom is one of the best. So welcome, Tom. It's great to have you back here. This is not your first time at the pub. So
no, it's not. Thanks, Laura, for the invite. Right, it was really nice to get that opportunity to join here today.
Yeah.
Thank you.
So, Tom, anything in your glass tonight you'd like to share with us?
A bottle of tonic.
Well, we thank you for joining us, Tom, and not least, I'd like to introduce my trusty co-host, Dr. Jeff Elliott. Jeff, this is not your first time here either. So,
No,
what's in your
not.
glass tonight?
Well, since I work remotely in my home office, no one can tell me not to have a cocktail while we're doing this. This is a take on the tequila sunrise called the Holiday Sunrise. My daughter came up with this over a holiday competition that we're doing this season. Started Thanksgiving, going through Christmas. And each of us has to come up with a seasonal cocktail, and we judge them, and the winner gets, I think, a prize. And I'm probably having to pay for that prize, but I haven't been told what the prize is yet. So
Yeah.
But yeah, she did a really good job, so I'm having it again. So
Good deal. Well, thanks for joining us tonight, Jeff. Now, I've been in a rut lately, and I think I mentioned that on my last podcast. I've been having Woodfords every time, and so I decided I was going to have something different. So I went and bought a nice bottle of Widow Jane. But that plan was foiled. Our pump quit working here at the house. We didn't know what to do, so my wife Mary says, you know what, our new neighbor, I've never met this guy, he's a retired plumber or electrician or something. I think I'm going to give him a call and see if he'll come over and look at him. I'm like, okay. I said, gee, can I pay? And he says, no, I won't take anything. But we had been, while he was there, we had gotten talking about bourbon, so he likes bourbon as well. So I tell you what, I got a bottle of Widow Jane that I'm willing to part with. So Mark, I don't know his last name. Mark's got my bottle of Widow Jane. So that's my story this evening. Anyway, all good plans, huh? Folks, thank you for joining us tonight. So I look forward to a great conversation. So cheers.
yeah, cheers.
Cheers.
The economics of feeding Reassure precision release choline. Reassure is fed during the transition period, and because it's fed for such a short period of time, it costs just $15 per cow, and yet the benefits will continue to generate income throughout the year. Cows fed Reassure produce 5 pounds more colostrum, which pays for your Reassure investment on the very first day of lactation. Cows fed Reassure also produce 5 pounds more milk per day, every day. That means after the first day, every day is payday. Invest in Reassure during the transition period and recoup your investment on the very first day of lactation. After that, you got it, payday. So Laura, I'm going to jump right into it. In 2024, you hosted a great webinar before the Real Science lecture series that led to some interesting conversations and questions. Can you please give us a quick overview of that webinar and help explain the topic of the role of the mammary gland in governing calcium metabolism during the transition period?
Sure, I'd be happy to. So for a while now, I've been working in the field of calcium by accident. I really am a mammary gland biologist and was trained to kind of think about things in the production of milk and how that impacts the mom, or the cow in this case. And I ran into some things that along the way that suggested that the hormone I'm interested in was regulating calcium. And so I started doing a bunch of experiments over the last 13 years. And lo and behold, I sort of do calcium now. And because I was trained by someone who really thought, Dr. Bob Collier, that there were things coming from the mammary gland that could manage what the cow was doing. And so I've been working on that and looking at that. And over the last several years, I've been working on how we can do things in the prepartum period, particularly to manipulate what's happening in the mammary gland to make sure the cow has adequate, not only endocrine status, but nutritional status, and calve in better, be healthier as a whole from a reproductive standpoint, a nutrition standpoint, like an immunological standpoint. And what we've seen across a few different types of studies we've done, manipulating a bunch of different things, was that it's really this drain of the mammary gland of the milk, of which it has lots of calcium in it, highest mineral content, was triggering all these mechanisms. And so what I went back in the literature and dug to a really cool paper that I think I mentioned in my talk, back when you could do fun things like remove organs and add stuff back.
Yeah,
And it was a paper Jesse Goff did where they mastectomized some cows, and it was about 2 months before parturition. And what they showed was that the cows could calve in and maintain normal calcium if they weren't making milk. But if they were making milk, their calcium, rapidly decreased at calving. So we kind of went through that, and we've been looking at different ways to improve that. And in my very physiologically manipulative experiments, I crossed paths at one point with Jess McCart, looking at a lot of data that Tom has put out from his lab. And really, we're seeing similar things from an epidemiological perspective and applied nutrition perspective. And what we were seeing that really supports this phenomenon. And it's not the only one that occurs. Like, you could say the same for fat and energy and different components that are being secreted into the milk that are having this, what we call negative feedback. And so that's what I've been studying now. And I've been looking at a bunch of different ways to manipulate that and try to describe what's actually happening, so that nutritionists and producers can make decisions better, knowing what's occurring and what's the best strategy for their farm.
Yeah, I was watching your lecture again today in preparation for the podcast, and I've got a little bit of an offbeat question for you. But recently, Dr. Van Amberg presented some data showing how the percent of protein and fat has exponentially increased over the last few years. And it got me to wondering, has there been any changes in mineral content, be that calcium or phosphorus,
Mm-hmm.
and how has that changed anything, if anything?
It's a great question. Calcium and phosphorus are pretty stable in the milk relative to protein or fat. And fat by far is the most variable, and protein being the second. But it takes quite a bit to change the amount of calcium or phosphorus going into the milk. The one thing I would say that you can consider, and I don't know that it's a fact, is that with higher protein secretion, you get more calcium and phosphorus because there's calcium and phosphorus attached to the protein that's being secreted into milk. So what we would call the bound calcium and phosphorus might be higher, but not necessarily the free.
Got it.
But going back to his question, it's one of the questions I'd written down that more philosophical, but thinking about what is normal, first of all, and has that changed over the last 50 years, and especially relative to where we are on milk production. So Laura, you're suggesting that calcium and phosphorus, it's not hard to change it, but it stays fairly the same. But these cows are producing a lot more milk than they were 10 years ago, much less 50.
Right.
So I just wonder, has normal changed?
So that's a great philosophical question, because it has, and I would say that, like, one of the things that's difficult with interpreting what we do now versus what even someone did in 1985 is huge, because they are producing more, the animals are different sizes, the feed is better. There's a lot to be said for what we do. I do think that normal has changed. I think there's been some adaptation of the animals to maybe be more hardy, should I say. But I think they're more efficient. Like, certainly they were more efficient when they were on BST, that's without a doubt. But even without BST, they're fairly efficient. I don't know tons about protein, so don't shoot the messenger or anything here. It's just like a thought exercise. But even if you think about some of the stuff they do with protein, they're always trying to change things. What I read anyway is like 2 to 2.5%. And I can't figure out like how much of a difference they're making in any which direction. It's hard to tell because maybe quality's better. I don't know. Again, not a nutritionist, but cows are doing really well and we've adapted them to do so. So I feel like normal has changed. And that's where I struggle with like absolute concentrations of things, because we live off of that. That's what's published. And sometimes I think we're saying something could be ill or not well, and she may be fine.
I think normal has changed, right? So, I mean, but I also think it's changed for the better.
Mm-hmm.
We're focusing on issues differently, right? So, I mean, this goes back even kind of my time, right? But I mean, clinical milk fever rates at 5 to 10% of cows calving, right, probably was not unusual. I know for sure that, when I started, back 2000-ish, right? I mean, herds, 6 to 8% DAs as a percentage of fresh cows on a herd, that was pretty typical, right? And we accept none of that anymore, right? So I do get occasionally we'll have, Laura and I have some academic colleagues will say that, well, we haven't made any progress in transition cow health and things like that. And I quite firmly dispute that because we know at the farm level, indeed, we have.
Well, just look at somatic cell counts.
Yeah.
Yes.
I mean, anything. Our farmers, supported by our agribusiness, allied industry folks and university folks and things like that, they made tremendous progress.
Right.
And it shows up in not just health, but also in milk yield and reproduction and all that stuff. And all this stuff is multifactorial. But, 25 years ago, Laura, probably nobody was thinking about subclinical hypocalcemia.
No.
The 80s or 90s, right? It's
Mm-hmm.
and that's now
Absolutely.
where the focus is. And if you told me 15 years ago that there'd be as much calcium biology research, epidemiology as nutrition as we've had since then, I would have been like, oh, we figured calcium out a long time ago, right? And
Yes.
it's astonishing what your lab has been able to unravel from a mechanistic standpoint and otherwise that really has supported, some of our ability to kind of get this right out there.
I think so. And I can remember the first grant I submitted, someone said, maybe it was you, Tom. I'm just kidding. Maybe it was. But like someone actually put in there, we don't need to study calcium. Like it's done. I was like, oh, okay. Maybe it is. I'm wrong. But it's been fun because I think, like, to what Tom points out, is it's been a synergism of producers, veterinarians, academics, like really trying to say what is happening and before making any like very definitive claims and how we can adapt stuff better. But it's amazing how healthy I think a lot of our animals are.
So, Tom, you talk about it being multifactorial. Is it genetics? Is it management? Is it our understanding?
Yes.
Yeah, all of it. Yeah, I knew you were going to say that. Wouldn't put an emphasis somewhere?
Well, I think probably depends a little bit on what it is, right? I think for production traits, components, things like that, genetics is certainly in there as well as the management. I think for the things that we're talking about here today, much of it's the management around it, dietary and otherwise, right? Then helps these cows make it through this period better than they used to. So, not to say we don't have opportunities still, right? Because we sure do, and there still are. And Laura, you did a nice job. There are still questions out there, right? But I think our understanding has come a long way, and our ability to manage calcium metabolism has come a long way as well. So,
I'd say too, I think that like digging deeper has provided more opportunities for farmers to use practices that work best for their management system rather than only being able to tap into like one thing. Here's the one thing you can use and try to shove a square peg into a round hole kind of situation. Whereas like we have such a large range of size, type of farm, milking system, management practice, people working there. And it really, one thing my colleague Milo Wiltbank always says, like double off sync works, but they have to give them the shots when they're supposed to, right? Like, and I think that could be said for pre-fresh nutrition too, or anything else. Like, so if you have more tools in the toolbox, you can use the one that like the people at your facility and farm can implement correctly, and know how to handle the animals, the response to that more efficiently, because it's not a one size fits all by any means.
Yeah, talking about tools, you presented some data during your presentation on some of the interventions that are being used. Can you talk a little bit about that data? Maybe what your
Sure.
recommendations might be?
Sure. I think, kind of the classic things that are out there, like the really like low input kind of strategy would be like a low potassium diet. And there are folks who still do that. And that's good enough for them, and they can do it consistently and manage their herd pretty well from a pre-fresh standpoint. The other two would be like clearly negative DCAD diets work. There's a lot of them. It's if you have some kind of negative DCAD diet, metabolic acidosis, and your urine pHing works really well. If the cows are eating it, if the diets mix well, all in all, those things work. You can do it for a long period of time without having any issues with the cows. So, like, depending on what your penning strategy is pre-fresh, like, that might be the easiest. There's a newer technology to the US anyway, was being used more in Europe, but that would be the zeolite clays, both of which Tom and I have done a little bit of work with. And that is a binder that binds 2+ ions, so like magnesium, phosphorus, calcium. It was largely thought that it was primarily a calcium binder. My data, and I think more data now, suggests that it probably works primarily through a phosphorus reduction mechanism. Um, and that is a more like acute treatment of 10 to 14 days prepartum, which, if you can do it, appears to work well for calcium purposes. But you can't really feed it for an extended period of time. That's appears to be somewhat contraindicated. And so they're still working out some of the information related to that and application in the US, because all the data prior to Tom's study was performed in various situations in Europe. So um, I've heard like some of the systems where they are like grazing and they're maybe only milking one time a day, that might be simpler for them to implement. But I think I've heard from producers that DCAD works really well if they can do it. The zeolite clay will work really well if that works for them. But again, it gets back to your grouping strategy, compliance by the staff on mixing and urine pHing if you're doing DCAD. And, those kinds of things become important. But what we've shown is that you have to be looking at things from a couple of different ways, because the clay is a phosphorus driven biology. And so management of cows is different, I would argue, compared to management of cows like in a metabolic acidosis, which is largely a calcium driven biology. And so I think there's questions from veterinarians surrounding low phosphorus, what that might mean long term, and some of the outcomes in post fresh cows. Like if you do have a cow that goes down, she's almost certainly low phosphorus and not low calcium. And I don't think people were thinking about it that way initially. And that's why the work we do is really important, because in real life, in real time, the vet or the farm staff has to make a decision of what to do. And if you give a cow who's low phosphorus calcium, you can have rebound milk fever. So those kinds of things are really important. And so I would say from a pre-fresh strategy, those are the two like tools we have in the toolbox beyond managing potassium. But I'd be curious to hear what my colleagues have to say on that.
Yeah, I mean, Laura, you hit it really nicely, I think. I mean, of course, low potassium diet is just partway on the way to low DCAD, right? Because it's one of the cations you're managed. Um, so Laura, I did also watch I'd watch your webinar shortly after I was invited to join you,
Mm-hmm.
and I also watched it at 5:30 this morning for this.
What a way to wake up!
that and a good cup of coffee was a nice way to start the day. But anyways, I think one of the things when you present this stuff, and I think it's really interesting because you refocus us a bit, right? Because a lot of us think about metabolic regulation, the transition cow, and it's just like all this stuff happens in all these other body tissues to just like throw stuff at the mammary gland, and you kind of think of the mammary gland as the receiver in this case. Yet you do a great job of kind of reminding us that the mammary gland is actually a very active participant in that regulation and calling the shots. And I think that's fascinating to it kind of takes me back also to some mutual mentors of ours like
Mm-hmm.
Bauman and of course your primary Bob Collier, people like that, and really interesting. And I think one of the things that also struck me, when you're listening to your discussion on calcium balance and lactation. And I guess I either didn't realize or didn't pay attention, but I mean, you say these cows are in negative calcium balance through 150 to 200 days in milk. That's amazing.
Actually, yeah. It's quite it's quite like overwhelming at times. And every time I've spoken with Jess, and I just saw Ralph Nevis a couple weeks ago at Purdue, and I'm like, man, we need to get like a machine that we can put the cow through so we really know what her bones look like. Because she's very resilient, and so are all like mammals when they're lactating. It's quite impressive that they're able to get to this like neutral and reaccrue what they need. And that biology is always stuns me. That's kind of one of the conserved phenotypes of like mammals in general. And you want to make sure they're getting enough calcium, but there's this compensation, like a lot of different things that's occurring from her directly as we keep pulling milk out, until she can hit that peak phase, and start to shift gears again. So,
Because that's not a
yeah.
lot of time between that time and when she calves again, right? I mean,
Mhm.
it seems like she's got a. I mean, I think you said there's like 8.5 kilos of total
Yeah.
calcium in bone. Is that total in bone?
Yeah.
And so, how
Yeah.
much? What does she go down to? Do we have any? We don't really know.
No, I saw some really, really old papers, but they were largely like in sheep. But I haven't really seen a good one in cow that can in bovine that to talk about that. And this is why Ralph told me I can get like a 600 kilo cow into the horse micro CT, and I was like, well, I don't know. That might be still too small, Ralph.
Mm-hmm.
But I think I'd really like the opportunity to kind of solve that, because maybe she's completely different, or maybe it's not as robust, but we don't really know. And the only way, like we've thought about, is taking like a biopsy from the pin bones to see if we can get at least turnover within the tissue, because it's really hard to decipher in an animal that size, without ashing her, which is not what I want to do. So,
From a nutrition standpoint, I mean, I occurred to should we be feeding more calcium in lactation?
possibly.
I don't know.
Intuitively, that's what it sounds like.
Yeah.
Right.
Or,
Yeah.
or is some of this just hardwired into her regulation and metabolism, where she is just going to, her bone is just going to do this regardless of what else we do? And you think about things like 25 hydroxy vitamin D and things like. I mean, boy,
Yeah.
it's
Well, I'd say in humans, like they'll lose up to 10%, sometimes 15 if they're breastfeeding only for the first 6 months. So it's pretty like hardwired that she'll keep pulling it until they get to parity, right? And then she'll start to like, I don't know if it's that they rapidly increase absorption and that allows for more storage, if it shifts the hormonal signals. But that's one thing I'd like to do as well, and be able to do it in a live animal accurately. Because that's the other part too, is the gut part, right? Trying to understand what's happening in the gut in real time, because that's a difficult thing to get at without using like calcium 45 or something, which they are not in favor of you doing these days. So trying to understand that better is like, what's the rate of absorption within the gut, and like that, in relationship to her endocrine status and her milk production. I'd really like to do that and be able to compare it across stage of lactation. So like the late pregnant cow, the early lactating cow, and then the cow that's like at 150, 200 days in milk, and try to see if we could put that puzzle together.
So, Laura,
Mhm.
I guess I've kind of accepted, as I came into this industry, that multiparous cows are more prone to milk fever. That's what we learned. First calf heifers don't. But why, we just accepted it, and now it's making me think, why is that? Is there less ability for those cows for bone resorption, or is it in later lactation, when the calcium demand's not so great, they're not getting enough calcium back into the bone, that they don't have it for the next lactation? Or is it
Right.
just the biology we don't understand?
Right. Those are all great questions. I don't have a definitive answer, but I don't know if that as they age, like they're just not as effective in the absorption of the nutrients, or if there's also something related to, because we do know females have a harder time with their bone because of estrogen. And so I don't know how estrogen declines like overall in a cow, like, or in her lifetime as it compares to a human. But it's one of the things that they've seen like in humans where that have offspring later in life. They can have issues with bone turnover, and they'll see fractures sometimes in those lactating women because their density is lower because their estrogen levels are lower. On the same side, like, and I wonder this because we've learned too what you just said, it's what I learned. But in those growing animals, is there some kind of missed opportunity there too, that we don't know what's happening in them because they're still depositing bone? Because they see those also occur in, like, say, teenage um, women sometimes as well, that when they have a baby and they feed, um, do breastfeeding, they'll have problems as well because they're still depositing bone until they're like 25. Um, so I don't know if it's an endocrine thing, it's an efficiency of their gut, potentially their absorption, or if it's also like, get real technical, stem cell sort of thing, kind of like you see in the mammary gland over time as lactation goes on, or you go through subsequent lactations. I think it was like Tony Capuco that did this work, but like they have less and less stem cell populations in their tissue to have regeneration of the tissue. So I don't know if that's a thing too. But there's these like disparate ends of young and old, and then there's the ones in the middle that do mostly okay.
It's intriguing for sure. I even had a nutritionist call me one day, and he asked, and I think you and I talked about this at the Cornell conference. He asked me about vitamin K2.
Oh, yeah, you did.
And I was like, I don't even know what that is. And he said, well, Balchem makes it. And I was like, well, we make it on the human side. Scott, I don't know if you're aware of this.
Yes, I am.
But
Yeah.
apparently one of its functions, I think of vitamin K as blood clotting, but K2, vitamin K2, part of its function is getting calcium into the bone. It expedites that. And he was wondering if that, could we feed some of that late lactation to help just basically longevity of the cow as well, not just about calcium mobilization,
right?
Better bones. So it was, yeah, I mean, there's, we don't know what we don't know.
Sounds like a research project.
Exactly.
So
exactly.
Laura, so one of the interesting areas that we, kind of get into, right, is, what farms do or what producers do at the farm level to give calcium, right, in different ways, shapes, and forms. And there's some older data, not that old, I guess, where, folks gave, 500 mils of 23% calcium borogluconate IV to cows that were just subclinical, and they got a big spike in calcium and then some rebound hypocalcemia after that. And I was really interested to when you talk about Megan's study there, right, when you're basically just trying to use calcium gluconate post calving. But what was interesting in yours is that a farm gives that at 500 mils in a relatively short period of time, whereas you guys spread it out over like 20. I mean, I think it was 100 mils an hour for
about that, yeah,
24 hours, and yet they still crashed. Essentially, when you guys quit doing that calcium. So I'm curious what your, we've been, Jess and I have both been pretty, one, when a farm has milk fever issues, like really trying to get to the bottom of, okay, what is that farm actually doing beyond what might be happening with the diet, i.e. calcium administration. But, I mean, I find that really interesting that even in your case, where you spread it out, they still crash pretty hard, it looked like.
Yeah, and this gets to the timing, and timing is everything. And that's where I showed her that data one time when we were both like on a speaking thing and we were talking, and then she showed me some of hers and Roth's data about the classification, and that was where this really became interesting because we were trying to keep the cows normal calcemic for the right after calving. I think during that time, most people were trying to take a blood sample within the first 24 hours of calving and making decisions. Subclinical, clinical, this is what our population looks like. This is what we need to do. So our goal was to really say, like, these, and what my hypothesis has always been is that you have to have a decrease to trigger negative feedback. That's how negative feedback works. So we wanted to keep the cows so normal calcemic during that first 24 hours. And so we were just adjusting our infusion rates based on the cow's response to the calcium. And what we saw was, because she never decreased, or none of them decreased during that first 24 hours, when we stopped giving them calcium, they had their negative feedback response. And it was like, we need to go down because we need to reset everything to come up to what's appropriate. And we saw that even in the cows that were receiving the negative DCAD diet. So we prepped them to calve in better. And then we did that, and then that kind of erased our negative DCAD diet response that we saw when they were just getting control saline. And so to me, what that says is that they need that time. And so when people ask me what to do, and I'm like, try to get to 48 hours before you take a blood sample. Please try. Like, if you really are itching to take one earlier, wait till 36, but like ideally wait for 48 hours, because calcium, and I think I showed this in the talk, is really a combination of when you decrease your calcium, how far you decrease the calcium, how long it stays decreased, and when it decreases. And that was exactly what Jess saw in her classifications of like the delayed, the persistent, the transient, and the normal hypocalcemic animals. And when they were classifying cows in that way and looking at these clinical outcomes of DA, or, take your pick, it came back to this like 48 hours time frame. And because you could take a blood sample at 12 hours and it might be normal, but she hasn't gone down. And if she goes down later on in those cows that they classify as like your late delayed hypocalcemic cows, you would miss those. And similarly, you might get the opposite, treat a cow that you didn't need to treat because she was at her nadir and coming right out of it, right? And of course, it all depends on what your cut point is, which no one seems to agree on. It depends on what your outcome variable is. And so that has been what I tell folks when they call me, and I've told them, like, if you can wait, unless a cow is ill, like, let her be. And then if you think she needs a bolus or an IV, like, sure. If you know she had milk fever last lactation, or maybe had a DA or RP or some other combination thereof, maybe then give her a bolus too. Or if she's like a crazy high producer in her fourth lactation, she could probably use it. But in general, if you could leave them alone for 48 hours, they'll probably be all right, unless you're having bigger issues. And so that's what I try to tell people when they ask me for advice, which is here or there, not super often.
Yeah, and it's interesting. So again, people that do some of this, they do it with good intention because they think,
Totally.
okay, these cows, I mean, their heart's in the right place, right? They think, okay, cow needs calcium, therefore I give calcium. And yet, and there's at times it's like, I don't know, it takes a while in the discussion. Sometimes they're reluctant to admit what they're really doing. And so you got to kind of keep digging and digging and digging. And then it's like, oh, okay, yeah, all right. So Jess and I have had a few of those experiences together. It's also, pull a veterinarian in, the nutritionist in. Of course, you get milk fevers, nutrition is blamed, right? So regardless of what we're going at the herd level, and so it's why you just got to try to understand it all. But it's really, really insightful. So
I'd say too, and the other thing we learned and why I think studying this area is important, or any of these really metabolic type health issues and understanding the physiology, is you can't give calcium to a lot of those exolite cows, because if they go down, I don't know what you guys saw with Allison's study, but they're usually low phosphorus cows. And unless you have a blood phosphorus, you don't know that. And so you give them the calcium, and then a lot of times they rebound, because what they really needed was phosphorus, and their calcium was just fine. At least that's what we saw in our experiment. And we had to hold the farm staff back. I had it explicitly in my animal care protocol, run the ISTAT before you give a treatment, because you could be giving them the wrong thing. So
yeah, we've done that at times. In that particular study, we actually didn't have to treat anybody, so
that's great.
We really never really bring into that issue of, okay, what do you do with her?
Mm-hmm. So. Yeah, I think we had a couple, so it wasn't a lot, but we had to take a couple off our study because they just gave the calcium, and lo and behold, their calcium went super high. And it was a phosphorus issue. And so I think that's why our science is super important for the industry.
I was also curious, you had that study where you did the prepartum chelation, right? The prepartum EGTA. I think it was 6 hours a day for the last 7 days, if my notes are accurate from this morning.
Good job.
I saw they needed more EGTA as they approach calving to really chelate that calcium. But I was curious I didn't look at I don't know if that's fully published or not. But colostrum yield in those cows. What does something happen if you're chelating?
Were they different?
Yeah, were they different?
They weren't on this study. It's not huge, right? Because we're doing highly manipulative things, but we didn't have any colostrum differences. What I've learned, and the purpose of doing that was to make these arguments that all these things work differently. If you're manipulating calcium, it does one thing. If you do manipulating phosphorus, it's another. If you're dealing with metabolic acidosis, that's something else. And they're all to the same endpoint, but understanding what each is doing would help you manage cows better. And what is one versus the other. But when we were doing this, we were focused on understanding specifically what was calcium doing from this standpoint. And I thought we were going to have to use not as much EGTA as we did. The early lactation cows that we've done EGTA treatments with, they are very resistant to that calcium. And I think, it's an effect of lactation. And so I suspect at this period of time, because of the amount of colostrum being pulled together and the higher protein concentration of colostrum, that there was probably significantly large draw at that period of time. Plus whatever the calf needed as well on top of it. And I believe that's largely why they were more sensitive than even the early lactation cows. But it was just another indicator that the mammary gland is no, it is for me. And cow, you stay okay, you stay good enough. But they were really fighting with each other on trying to take it away from her, because she wanted it, for partitioning, again, for the calf and for her milk.
Yeah, I think that happens with a lot of nutrients, right? Mammary gland gets the priority.
Yeah. So I don't know. It's kind of funny. I didn't realize I was saying anything too crazy, but I went to speak at Cornell the first time, I think when I was in Syracuse, in 2014 or something. And Dale was in the audience, and he's I am so glad you're saying this. this? And I was like, am I that radical? Like, this was the first thing I learned from Bob. But it's definitely a paradigm. Like Kevin Harvatine would be another that I think very much thinks from that perspective. But when you read all this stuff in all these maternal species, it is quite impressive what these mothers can do. And that's really like the basis of my science. So
Yeah, kind of a question for both of you. Laura, you started off saying that, in the very beginning, people said, hey, we know everything about calcium. And obviously, we've talked about a lot of different things today that we don't know. Well, where should the priority be in at least the near-term, mid-term research that needs to be done in this area?
In calcium?
Yeah.
I think that there's a lot of opportunity still yet to understand, for one, because there has been more uptake of Exilid and it's a newer technology in the US, like understanding how that works biologically, because DCAD is really, tried and true, but it wasn't at first either. And it took some time to get there. And so I think we should continue to be rigorous in our quest for understanding that. I also think that personally, because I'm interested in it, is this understanding of like the gut, what's happening in the mammary gland and in the bone in an actual cow, that, in a Holstein or a Jersey, but a dairy cow, and understanding that biology better. And like, I have some thinking of ways to do it without, costing a vet school probably many millions of dollars to buy a very nice fancy machine, but some other thoughts of how we could maybe get at that, like by measuring foot health and stuff, because those are high stress points for cows, like different ways we could maybe answer some questions. But I do think those are really important, and understanding in general, not just calcium, but I think in general as nutritionists and physiologists, that we should look at things at these different components of lactation, because a lot of times our studies are focused in the easiest animal to handle because they're more in control. But like, data is so different in these animals in a pre-fresh cow than it is a mid-lactation cow. And, we have a tendency to extrapolate because that's our best knowledge, right? And I think more work should be done in those because lactation is very dynamic.
Yeah. Tom, from your perspective, what would you say?
Yeah, a couple of things, right? I think from a field practical perspective, in both Laura's study and our study back in Binder, and actually when we did ours, it was all about calcium binding, right? There was no discussion of almost anything else, or there was like a bit of, well, maybe phosphorus is in here somewhere, but we were both at 500 grams per day. And that at the time we did it was like, that's it, period, end of story. Doesn't matter what else the diet looks like, things like that. It was just that. And now both companies that are marketing these binders, they're now relating it to a certain proportion of grams of phosphorus, which brings them down into the 300 plus or minus range, right? So versus the 500, and so nobody's really done that work. Nobody's looked at these things in one group dry cow system. I also share the reluctance to feed them for longer than the standard close-up period, just things like charted waters. I think from a more scientific standpoint, accounting for where all the calcium goes, right? It just feels like we don't have a great understanding, either prepartum or postpartum, of, because again, you get a little bit of calcium in the urine, but not a lot. You definitely get some in the feces. Obviously, you got some going in the bone or milk, but I don't know. When we try to do some rough math, it's not always easy to account for where things are going. It's got to add up to. It's got to add up
One.
to 100%. Got to add up to. So I don't know. So there's still stuff here biologically to kind of sort through, I think.
Yeah. Kind of circling back to something, Tom, that you had said earlier, and I think Jeff had alluded to as well, is those they don't start, they don't reach energy balance until later lactation. And do we know if they are depositing enough calcium back? And if not,
Calcium balance, not nutrition balance.
yeah,
Yeah.
I'm sorry,
He's
yes,
got
calcium balance. Yeah. And then how do we determine that? And then how do we improve upon that? You kind of mentioned nutrition, or is it, Jeff mentioned the K2? That is kind of intriguing. We
Cool.
do have that. I have no idea whether or not, I think it's expensive. I think that's what I've heard, but um, it seems to me that might be an area as well. Sounds good. Looks like the girls then just flash the lights. That means it is last call. And so with that, what I'd like to do is ask each of you, let's go around the table and what are just a few takeaways that you'd like to leave with the audience tonight? And Jeff, if you don't mind, would you lead us off?
Our last call question is sponsored by AminoSure XM Precision Release Methionine, the next generation in amino acid balancing. With AminoSure XM, you can save up to 5 cents per cow per day on your methionine investment. Try it today and receive an additional 2.5 cents per cow per day savings with Balchem's limited time rebate offer. Contact your Balchem representative to learn more.
Yeah, certainly. Well, I'd love the reference, because I'd forgotten about it too, that it seems like 20 years ago I'd heard that. Oh yeah, we know everything there is about calcium now. We're solid there. And it goes back to the old cliche, we don't know what we don't know. And there's still a lot to discover. And we've seen that with kind of the zeolite products, new products out there. Are there other ways to, skin the cat? So I think it's going to be an interesting few years in calcium mobilization, but a lot of other things keeping up with milk production and the components that we have. So,
okay. Thanks, Jeff. Tom, thanks for joining us here again at the Real Science Exchange. What thoughts do you have for us?
Yeah, I just I've got a huge amount of respect for the work that Laura and her group have done to help us unravel some of these things. Again, Jess McCart on the again on the epidemiological applied calcium treatment side, right? And other groups have been in the mix too on the nutrition side. Laura, just a quick comment. My group doesn't often complain about sampling schedules, but if they ever do, I'm just going to point them to one of your papers because I think from a rigorous and frequency of sampling standpoint. So, but no, just fantastic work and a privilege just to come on today. So thanks, Scott. Thanks, Laura and Jeff.
Yeah, thanks, Tom. Laura, any final words for us?
I just want to thank you for the opportunity to talk about it. I know I'm a pretty intense physiologist, but I think I was trained to be curious, and that lactation is a cycle, and that we have to really think about that and how the mammary gland is directing the show. And so I like to approach things from that way. But I've been fortunate to learn from a lot of cool people and had a lot of great support. And it's been fun as a basic scientist to cross paths with, epidemiologists and see your work really have impact. And that's been a lot of fun in getting to know Jess and Tom and Sabina Mann too. And we've done some cool things together. And Tom, I buy them a lot of pizza and cake and throw a lot of parties. And I often go out there and sleep at the barn with them if they let me. But I've been really lucky with an amazing group of scientists over the years. So they almost always are game, which is a surprise because the sampling schedules are brutal.
One other thing I would add, I was thinking about is, the last few years we've talked a lot about the longevity of the cow and how many lactations are we getting. And I think what Laura's doing is going to be key in that, aside from just, milk fevers, subclinical milk fevers. It's going to be key in that.
Yes, great point. So, Laura, I'm not an intense physiologist, and admittedly, this topic was a bit intimidating to me, but I've got to tell you, it's been enjoyable and understandable for me, and appreciate the two co-hosts here, Tom and Jeff, for coming along and helping out. This has been a great podcast. I've enjoyed it, and I want to thank you guys for, spending time with us and lending your expertise and knowledge to the industry. And to our loyal listeners, as always, we really appreciate the fact that you come here and spend time with us. We hope you learned something. We hope you had some fun. And we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email at anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars takes place on the first Tuesday of every month, with the top research and nutrition topics that will impact your business. We also include small ruminant, monogastric, and companion animal-focused topics throughout the year. Visit balchem.com/realscience to see the upcoming topics and to register for future webinars. You can also access past webinars and search for the topics most important to you.